Hello, and welcome to the latest Technology Networks webinar, Profiling Age and Body Fluid DNA Methylation Markers. I'm your moderator, Dr. Karen Stewart, Senior Scientific Specialist for Technology Networks, and I'm excited to be hosting today's session. We have a fantastic presenter, Dr. Zaki Ewan, who will be sharing valuable insights with us. But first, I'd like to introduce Shionika Monhanta, the Strategic Marketing Manager in Epigenetics from Oxford Nanopore, to give you a brief introduction to DNA methylation and methylation detection.
Hi, everybody. My name is Shionika Monhanta, and I'm the Strategic Marketing Manager for Epigenetics at Oxford Nanopore Technologies. Today, I wanted to give you a brief overview of what methylation is, why it's important in studying various human diseases, as well as a short introduction into how Oxford Nanopore's methylation detection works. So methylation is a central mechanism in human biology. In DNA methylation, a methyl group is added to the cytosine base. This doesn't alter the base DNA sequence, but can have a big impact on how our genes work, as well as how our cells function. It can influence gene regulation, cellular development, and genomic stability. But sometimes methylation can go haywire, and this has been linked to several diseases. In hypermethylation, genes are usually silenced, which has been associated with certain types of cancer. On the flip side, there's hypomethylation, which can cause genes to be overexpressed. And this has been associated with autoimmune disorders and certain neurological conditions. In mammals, the most prevalent type of methylation is 5-methylcytosine, commonly called 5-MC. This is a foundational component of DNA that plays a significant role in regulating genes within our genomes. Now let's talk about CpG sites. CpG is essentially a shorthand form of cytosine followed by a guanine base. These sequences tend to cluster around in regions called CpG islands that are commonly found in gene regulatory areas. It's worth noting that the process of adding methyl groups like 5MC to the CpG sites is intricately linked to gene silencing, essentially turning genes off. So in a nutshell, understanding this interplay between 5MC and CpG sites helps us understand how our genes are regulated, and is a crucial piece of the puzzle when it comes to figuring out the mechanisms of various diseases and finding potential diagnostic markers. Let's look at a few examples of how this happens. So in cancer, hypermethylation of CpG islands in the promoters of tumor suppressor genes leads to increased 5MC, causing gene silencing and uncontrolled cell growth. In neurological disorders, changes in 5MC levels are noted in genes linked to neuronal function and survival. This plays a key role in the development of various neurodegenerative diseases. Similarly, variation in 5MC levels in genes associated with vascular function and lipid metabolism have been linked to several cardiovascular disorders. Thus, studying methylation patterns in DNA and RNA can offer valuable insights that could lead to potential diagnostic and therapeutic interventions. Traditional methods for detecting DNA methylation rely on either chemical or enzymatic modifications. One commonly used method is bisulfite treatment, where unmethylated cytosines are converted into uracils, thus allowing the differentiation between methylated and unmethylated cytosines. This process is usually followed by PCR amplification and then sequencing to analyze the methylation patterns. In contrast, nanopore sequencing can directly detect methylation status of both DNA and RNA without any additional library prep or sequencing. Oxford Nanopore sequencing is also read agnostic, meaning it doesn't matter if your DNA is 20 base pairs or 4 million base pairs. This versatility allows nanopore sequencing to sequence CpG islands, providing a comprehensive and precise analysis of DNA methylation patterns across diverse genomic regions. Nanopore methylation detection can call all base modifications. So this includes 5MC, 5HMC, and 6MA in DNA, as well as M6A in RNA. This includes complex and repetitive sequences, You can phase the methylation with nucleotide level precision. This approach is also adaptable, that supports both targeted and genome-wide detection, and can be scalable based on your throughput needs. All this data can be acquired real-time and can be adopted or optimized during a run. Now I would like to introduce Dr. Zaka Yuen from the Australian National University, and she will be walking through profiling age and body fluid DNA methylation markers using Nanopore adaptive sampling.
Thank you, Shanika. It's now my pleasure to introduce our speaker, Zaka. Zaka is a postdoc in Professor Eduardo Ayra's lab in the Australian National University. During her PhD in the same lab, Zaka benchmarked nanopore methylation tools and explored PCR-free nanopore targeting techniques, including Cas9 enrichment and adaptive sampling, investigating the use of MinION for forensic SNP genotyping and CpG methylation profiling. Currently, her research continues to focus on utilising nanopore sequencing for advanced methylation studies, particularly in the context of cancer progression. After the presentation, we will have a Q&A session, and we encourage you to submit questions at any time during the presentation. To do so, simply type your question into the box on the right-hand side of the screen and click Send. We will do our best to address as many questions as possible within the time available. If you encounter any technical difficulties during the webinar, please click the chat box on the right-hand side of the screen to request assistance from our support team. So without further ado, I will now hand over to our speaker, Dr. Yuen.
Thank you, Karen, for the kind introduction. My name is Zaka Yuen, and I'm a postdoc at the Australian National University. Today, I'm excited to share insights into our work on profiling DNA methylation for age and body fluid markers using nanopore adaptive sampling. DNA methylation is one of the epigenetic modifications crucial for regulating genome stability, gene expression, and mammalian development. It also plays significant roles in disease progression and aging. Among various DNA modifications, 5-methylcytosine, 5-MC, stand out as the most frequent observed in relation to gene regulation. It is also referred to as the fifth base of DNA and occurs at the cytosine bases within CG dinucleotide of the eukaryotic DNA, known as CpG site, which is also referred to as the CpG methylation. The transformation from cytosine to 5-MC is an enzymatic process that adds the methyl group to the fifth carbon position of the cytosine ring. And this reaction is facilitated by DNA methyltransferases, with several types of the DNA methyltransferases playing roles in this intricate process. CpG sites are frequently found in the large clusters known as CpG islands, which are CpG-rich regions located in many gene promoters. These sites are instrumental in controlling gene expression. For example, the presence of the unmethylated CpGs in the gene promoter region is associated with active gene expression. DNA methylation is a fundamental mechanism in the epigenetic landscape, influencing gene expression and thus impacting development, health, and diseases. The DNA methylation landscape is dynamic, which is continuously reshaped by a combination of environmental conditions and the aging process. It undergoes changes during early development, cell differentiation, tissue development, and aging. As you can see here, DNA methylation undergoes significant alteration during development, started from the germline and early embryo stages, where extensive demethylation occurs to reset the epigenetic state, followed by the establishment of methylation. The figure on the right is inspired by Waddington's landscape metaphor, which visualizes the journey of cell differentiation, starting with pluripotent cells at the top and ending at the lower point when a differentiated state is reached. And DNA methylation plays a crucial role in shaping the epigenetic landscape and determining the identity of the cells during development and beyond. Since DNA methylation varies across cell types and tissues and throughout an organism's lifetime, it has been identified as one of the most promising biomarkers for predicting age and identifying body fluids in forensic science. Estimating an individual age through DNA methylation is useful in several areas. From a forensic point of view, the ability to accurately predict chronological age of the donor of a biological sample can provide relevant info in order to guide police investigation in cases where no suspect or matches in the DNA database are found. As human age, there's a global decline in DNA methylation, and this process, known as hypomethylation, occurs across the genome and is a characteristic feature of the aging process. However, for age prediction, our focus often narrows down to specific CpG sites linked to age. The methylation level at this site may correlate either positively or negatively with age. For instance, there are two age-associated CpG sites. The red one here shows methylation increases with age, while at the blue side, methylation decreases with age. Also, identifying body fluids is important for crime scenes reconstruction. The detection of certain fluids like semen can indicate sexual insult. Several studies have identified unique DNA methylation markers for various body fluids, showing significant differences in methylation levels between tissues. Numerous DNA methylation-based age prediction models have been developed to infer an individual's age. There are two major types of these models. The first type, which are referred to as the epigenetic clock, involve the use of 100 of CpG sites. And this epigenetic clock adopt a more comprehensive approach, emphasizing the biological aging process, which accounts for variations across tissues and the maximum lifespan of the species. They can be tissue-specific, tissue-wide, or even applicable across many mammal species. The second type of the age prediction model is commonly used in forensics. There are tissue-specific and use small sets of the age-associated CpG sites, usually around 4 to 8 sites. It's important to note that the CpG site chosen for forensic age prediction models differ from those featured in the epigenetic clock models for several reasons. Forensic models primarily aim for precise age prediction of individuals, usually within a specific age variation range. Unlike forensic models, the epigenetic clock focuses more on the biological aging process. So not every CpG site linked with chronological age is also associated with biological age, and vice versa. In terms of technology, epigenetic clock often utilize microarray technology to analyze hundreds of thousands of methylation sites simultaneously. This is a traditional technique for assessing the methylation status of cytosines, but it relies on predefined probes that restrict the detection to known sequences. For forensic age prediction models, they utilize a broad spectrum of targeting approaches. Capillary electrophoresis, a traditional technique to analyze DNA fragments, is useful for specific methylation studies, but has lower throughput. Power sequencing, an older targeted method to measure methylation at specific loci, which offers a rapid and reliable result, but is limited in genomic coverage. Snapshot, also a traditional approach for genotyping and methylation analysis at known sites, is useful for specific target, but not for discovery-based research. Massively parallel sequencing, MPS, as you know, is a widely used technology today that provides genome-wide methylation profiling. Mass spectrometry is a specialized method for targeted analysis of known methylation sites, but is relatively expensive. And despite the diversity in the applications and methodologies, A common characteristic of these old technology is the reliance on the bisulfite conversion to detect methylation. So bisulfite conversion involves treating DNA with sodium bisulfite, which converts a methylated cytosine to uracil, while a methylated cytosine remains unchanged. The DNA is then amplified by PCR, where the uracils are converted to thymines, which allows for the differentiation between methylated and unmethylated cytosines. The drawbacks of bisulfite conversion-based method include a substantial DNA degradation, as the bisulfite treatment process is harsh and can lead to degradation of DNA. Incomplete conversion, which means unmethylated cytosines may not be converted to uracil, leading to false positive results. PCR artifacts, because converted DNA has reduced sequence complexity, PCR may preferentially amplify certain regions over others. Non-experimental protocols and complexity in data interpretations, since the converted DNA sequences become more similar, which complicate the alignment of sequencing weights to the reference genomes, especially in regions with high CG content. Another limitation of the current assays for age prediction and body fluid identification is that the DNA methylation markers for each purpose are detected separately, which increases the total number of assays required. To overcome traditional challenges in DNA methylation detection, Nanopore sequencing emerges not merely as an alternative, but as the preferred choice. Nanopore sequencing enables the direct detection of DNA-based modification with single nucleotide resolution. Its PCR-free nature eliminates the biases and artifacts typically associated with PCR. Moreover, it provides long reads, allowing us to target entire genes or regions rich in CpGs to profile numerous CpG sites comprehensively. In terms of methylation detection, it has been benchmarked against conventional bisulfite sequencing, which has become the new gold standard for methylation detection. With targeted nanopore sequencing, we can enrich for loci of interest, including age-associated and body fluid-specific DNA methylation markers in a single assay, which minimizes the number of sequencing runs, thus saving both time and resources. This targeted approach ensures high coverage data at desired regions. It is also more cost-effective and enables more efficient data analysis. The targeted nanopore sequencing approach that we used is adaptive sampling, also known as read until, where the nanopore sequencer can identify the target sequences and directly reject or accept DNA molecules during sequencing without specialized sample preparation. So a fragment that enters a pause will produce a sequencing data in real time, followed by rapid alignment to the reference genome to identify the target. If the target region is found, sequencing continues. Otherwise, the read is rejected by reversing the voltage across the pores, and the pore will sequence the next strand. In this way, unwanted sequencing data is minimized, and coverage can be focused on regions of interest only. So our study aims to corroborate existing forensic age and body fluid DNA methylation markers and identify potential new markers using nanopore adaptive sampling to investigating all CpG sites within the target genes. To do this, 3 types of the genomic DNA samples were used in the adaptive sampling runs. Firstly, the genome in a bottle reference DNA sample, HG002, was employed to evaluate the performance of our custom-designed target panel. Secondly, we used methylation control samples that were derived from human whole blood and enzymatically treated, resulting in a high methylated control with over 85% methylation and a low methylation control with less than 5% methylation. Lastly, we ran on 10 human blood samples. Blood samples were collected here at ANU from the 10 participants aged from 25 to 76 years old. For library preparation and sequencing, we used a ligation sequencing kit and the R9 flow cells. We then performed adaptive samplings on these samples using a custom panel comprising many markers associated with age and body fluid identification. The entire panel covered around 113 megabases in totals, targeting approximately 3.65% of the human reference genome. Our analysis workflow comprises the following steps. We utilize FASTFile files as input and perform modified base calling using Bonito. In this process, we specify the reference genome and the Ranmore model. So Bonito is an open source ONT base caller, while Ranmore serves as the core API for modified base calling, working in conjunction with Bonito by relying on the output of the base call processing step. Bonito then generates a mod.sam file, which is a SAM file containing alignments with base modification cost information. Subsequently, I convert the SAM file to the BAM format and then utilize modkit to further convert it to the BAM file, which contains number of reads and the methylation method of the CPG site. Although adaptive sampling has the potential to enable simple, cost-effective sequencing of different DNA methylation markers, but it is a largely untested technology used in forensic contexts. So the first thing we need to check is, does adaptive sampling work as expected? As I said, we perform adaptive sampling using our target panel on the well-known reference samples, the genome in the bottle reference sample HG002. The figure on the left here shows the distinct distribution of accepted reads, which matches our desired target size around 40 kb. In contrast, rejected read in red color here exhibited a sharp and narrow peak around 500 bases, which is the length selected to eject the molecules from the pore when they identify as not being from any target region. Additionally, there were reads with size ranges from 100 to about 2,500 bases, which the software could not classify. Therefore, these fragments were fully sequenced before any decision was made. Following the experiment, we used Guppy to rebase call the signal data and align back to the human genome. As you can see, on-target reads exhibit an M50 of 28.6 kb, much higher than the M50 of the 500 base pairs observed for the off-target reads. Lastly, adaptive sampling resulted in a medium 6-fold enrichment in coverage within the target regions compared to the off-target regions. This demonstrated the successful rejection of DNA fragments outside the target regions while fully sequencing the DNA fragments within the target regions. So here we can conclude that we can enrich regions of interest using adaptive sampling, which does not require any additional wet lab preparations. Next, we sequenced the control samples with high and low methylation to see if we could confirm the methylation status of the control samples using Nanopore adaptive sampling. Firstly, we observed overall methylation level that matched our expectations for each control samples. Specifically, the mean methylation was 93% for the high methylation sample and 4% for the low methylation sample. Secondly, our analysis revealed that even when only a single read covered the methylation course, the methylation level remains linearly unchanged, as you can see in the top right figure here. This consistency supports the reliability of nanopore technology in methylation detection, even with just a single read. But we acknowledge that the reliability of single-read methylation cores may vary depending on the sample's methylation heterogeneity, and this result primarily focus on the methylation control samples. We also examined the sequence contents around the CpG site and found an even distribution of the 4 bases around surrounding this site for both samples, indicating no sequence biases in our Nanopore sequencing predictions. Furthermore, we compare our Nanopore methylation data with the published whole genome bisurface sequencing data using the HG002 sample. We observed a good correlation as shown in the heat map with a Pearson's out of 0.86. To delve deeper, we randomly sample about 1,700 observations across coverage levels from 1 to 20, and we find that correlation values slightly increase with coverage. Moving forward, we aim to assess whether adaptive sampling can accurately determine the origins of blood in samples. Although we have access to only one type of tissue sample, which is blood, it serves as an excellent starting point. This will allow us to profile methylation at specific markers for body fluids and see the differences. Here, we use a recently developed panel developed by Lee, where they use 9 CpG markers to distinguish between semen, blood, vaginal fluid, saliva, and menstrual blood. This study used the snapshot multiplexed assay and relied on the presence of positive methylation signals at 2 CpG sites for all samples except for saliva, which was based only on one single CpG site, as only one site specific to saliva have been identified. And these 9 body fluid specific CpG sites were included in our target panel used in adaptive sampling. In this analysis, we examined the methylation level across the 9 sites in each sample. Each row represents a sample, while each column corresponds to a DNA methylation markers. The methylation level is indicated by the gradient of shade of blue. The darker the blue, the higher the methylation level. Additionally, the size of the dot represents the coverage, so the larger the dots signify higher coverage for the CpG site. As you can see, we observe significantly elevated methylation levels at the 2 target loci specific to blood. This indicates that the methylation measurement obtained from our 10 samples using adaptive sampling accurately identify these sites as specific markers for blood identification. It is also important to mention that the body fluid confirms the presence of positive methylation signals at the specific CpG sites, which is a relatively straightforward process. However, when it comes to age prediction, we need quantitative measurement of DNA methylation, which I'm going to show you in a second. So the next question we want to answer is, can we corroborate existing forensic age-associated markers with adaptive sampling? Before diving into the result, I would like to recap how we analyze the relationship between the DNA methylation and age. The plot on the left displays the 10 blood samples we have, showcasing the age and the methylation levels at the single CpG site. For our analysis, we employ simple linear regression to derive Pearson's R-squared and the p-value from the model. It is worthwhile to mention that it's common in forensic age prediction study to use a simple linear regression. Then utilizing the regression coefficient from the CpG site most correlated with age from each gene, the predicted age of individual is calculated for each specific gene. Then they select about 3 to 8 highest age-correlated CpG site for simultaneous analysis using multiple linear regression to construct a final age prediction model. We consider 7 different genes with a total of 32 CpG sites that are frequently incorporated in the predictive model used to estimate age from the DNA samples derived from blood based on the multiple forensic studies. As previously mentioned, we developed a linear regression model. Here you can see Pearson Owls represented in the gradient, where a darker salmon color indicates a better correlation. Adjacent to these, the bar plot displayed the mean coverage from different samples, with the error bars representing the standard deviation of coverage. Here we observe varying degrees of correlation, with the majority of CpG sites showing a positive correlation with age and an R value greater than 0.5. This suggests that the methylation level at these sites generally increase with age. And the site with the highest positive correlation with age was found in the gene EL0VL2 with an R value of 0.95. And several other sites within the same gene also show strong positive correlations. Considering the mean coverage across the samples, we achieved at least 10 reads, which is a good number for detecting methylation with nanopore data. With nanopore adaptive sampling, we streamlined the process of adding the new target regions to our panel, eliminating the need for the extra multiplexes typically needed with traditional methods. Here, we also include about 300 aging-related human genes from the aging gene database, as well as about 400 orthologous genes known for their age-dependent expression in mammals. Our next goal is to investigate whether this can lead us to discover potential new DNA methylation markers of age. Before jumping into the result, I first want to explain to you how we perform the meta-analysis to discover new CpG sites that could be associated with age. So after we aggregate the data from the 10 blood samples, we have got a total of 2,500,000 of CpG sites. Then we basically did what we have done for the forensic age markers. We performed a simple regression analysis on each site and generate correlation coefficient and associate p-values. Since the regression analysis is independent for each site, we then apply Stafford's unweighted method to analyze the statistical evidence across multiple CpG sites. This method is useful for meta-analysis, where basically you combine the result from multiple studies or tests to find an overall effect. So in this method, we first convert the p-value to the z-scores. Then we aggregate individual z-scores into a combined z-score, and then convert this combined z-score back to the p-value, which at the end, these are so-called the meta z-score and the meta p-value that we could use to compare across different CpG sites. With this approach, we successfully identify many CpG sites that exhibit highly significant correlation with age, as indicated by the red dots here, where we apply a meta p-value cutoff of less than 0.00001. This plot shows the genome-wide distribution of p-values on a log scale for all CpG sites identified in the data, plotted across the chromosomes. CpG site in red shows a relatively high significant correlation with age using this cutoff. Among the top sites showing the most significant associated with age, two highlighted in red here are located in an intergenic regions near a CpG site and a long non-coding RNA. Additional CpG site linked to age, shown in green color, meet the significance threshold with a meta p-value of less than 0.05. CpG site marked in gray are those identified in the aggregated sequencing data that do not show a significant association with age. Notably, we observed 5 CpG sites within the PLEKHG5 gene that demonstrated a high degree of association with age, marked in red color. There are also 5 CpG sites present in this gene, but only one significant CpG site was located within the CpG island. Furthermore, many other CpG sites in this gene, highlighted in green color, and using a matter p-value of less than 0.05, show an association with aging. And this gene involved in regulating neuronal cell differentiation has not previously been reported to have any known association with age-related changes or diseases. As mentioned earlier, Nanopore sequencing allows for sampling a great number of CpG sites by sequencing entire genes. CpG islands, typically found near the transcription start sites, are often associated with the regulatory function of genes and promoter regions. So we expanded our scope to include, but not be limited to, establish age markers targeting CpG sites within the CpG islands and additional candidates potentially correlated with age. For example, the ER-OVR2 gene is a well-known age predictor in blood, and the CpG site in blue color were previously shown in our result and incorporated into different age prediction models. Using a better p-value of less than 0.05, again, we identified 6 additional sites with a potential association with age in the ER-OVR2 gene, highlighted in the green color. Last but not least, we explore other CpG islands in our dataset to identify new sites with potentially comparable predictive values. For instance, within the CpG island in this ADCY5 gene, we discover some promising sites that could be useful for age prediction. To conclude, firstly, our approach is innovative. We use Nanopore adaptive sampling to simultaneously detect multiple forensic methylation markers for body fluid and age prediction. This is a game changer because it removes the need for any biochemical treatments of the DNA samples, streamlining the process significantly. Secondly, through adaptive sampling, we were not able to corroborate existing markers for body fluids and age, but also explore new potential regions. Our method also offers numerous benefits over traditional techniques. By adopting a PCR-free long-read approach, we have reduced laboratory preparation steps, minimized PCR artifacts, and increased the number of markers assays in a single assay. Importantly, this technology allows us to evaluate the genetic and epigenetic markers simultaneously. This means that we can analyze more targets like SDR, SNPs, and CpG site all in one go, making each assay more informative. Although we did include other forensic genetic markers in our adaptive sampling panel, the study of SNPs and SDR actually falls outside the scope of this methylation-focused study. However, we did a benchmark analysis to evaluate the overall SNP calling performance using all the SNP loci found within our target regions, and it shows a great varying calling performance with the H3O02 sample against the genome in the bottle, a ground truth. Importantly, no bisulfite conversion involved as we sequence the native DNA harboring the base modifications. We could capture and detect methylation from the nanopore signals directly. And with nanopore adaptive sampling, targeted nanopore sequencing shift from traditional wet lab manipulations to advanced dry lab computational strategies. This not only makes the process more efficient, but also more portable, opening up new possibilities for forensic analysis on the go. Nanopore sequencing has the potential to transform forensic DNA analysis. Few portable nanopore sequences like the mean iron could become all-in-one forensic DNA profiling devices. This would enable the simultaneous analysis of about geographical ancestry, kinship, phenotyping, body fluid identification, and age estimation of the DNA donor. So to conclude, our study was the first of its kind to use nanopore additive sampling to simultaneously detect DNA methylation markers for different forensic purposes in a single assay. And it has laid the groundwork for a transformative approach to forensic DNA methylation profiling. For more details on our research, scan the QR code on this slide to access our latest preprint. Nevertheless, there are some limitations to our study that merit attention and open avenues for further research and improvement. Firstly, our exploration into age markers revealed a mixed bag of results. Some markers demonstrated only a mild correlation with age, underscoring the variability across technologies and the need for more extensive and diverse sample sets. Secondly, the high DNA input requirement for nanopore sequencing present a major limitations, especially when dealing with low quality or degraded samples, which are all too common in forensic context. This limitation emphasizes the gap between the ideal laboratory conditions and the gritty reality of forensic work. Our study's scope was also limited by the lack of replicates, limited samples, and the diversity of our sample set. As for future directions, we will require larger, independent sample sets that span a broad spectrum of age ranges and biological sources. This will assist in improving the statistical power of the tested markers. In tandem, we anticipate advancement that lower the DNA input requirement for nanopore sequencing. This will make genomic sequencing more feasible for forensic samples. So with that, I would like to thank my supervisor at Wonder Iris, my collaborators, Cameron Jack, Dennis McNathan, and Runa Daniel, as well as all the lab members in the Iris group. I also want to express my gratitude to Karanina and Lachlan from the ANU BLF for their assistance with library preparation and sequencing, to Simon for providing the blood samples, to Simon's team for their work on DNA extraction, and to Ladine for preparing the DNA from the cell line. Thank you very much for listening.